Okay, so we'll just go around the room and everybody, like, you can read out the question and your answer and justify it.
Yeah. I don't have it with me. I didn't bring my laptop.
It's okay.
Okay.
I got these like first three for you.
Oh, that's good. Alright, so the first question we have, sorry, still chewing. What two systems are involved in the vast majority of feedback responses that maintain homeostasis? You have the answer as circulation and the central nervous system.
Okay, now justify the answer. Why is that the correct answer? Not just because they gave it to you, why is that the correct answer?
I have to be honest. This question threw me off just because of that chart that he originally had. He like circled everything, so I was confused. I mean, nobody else may be confused, but I was. So that was just me personally. I know he spent a lot of time talking about circulation, and of course, you know, CNS makes sense, but I feel like.
Well, the whole, I mean, basically everything in some way or another is controlled by the central nervous system. So the central nervous system was a given.
Right.
Yeah.
And then you just figure out, then there are only two answers for the central nervous system. So is it circulation in the central nervous system or is it central nervous system in the kidneys? So why do you think they chose central nervous system and circulation?
Well, I figured because if it wasn't sent via action potential, it could be sent like through hormones and stuff to effectors. Like if you needed to change something.
Yeah, so the kidneys are actually, kidneys are one of the effector organisms, but what they're affecting is circulation.
Right, and let's say kidneys affect circulation.
Right. So it all comes back to getting things to the tissues, and that requires central nervous system stimulation and signaling and circulation.
Well, without circulation, your tissue would die.
Pretty much. Yeah. I think we've all done the stupid thing when we were kids, put the rubber band around our finger and watch it turn blue and then gray and then go, maybe I shouldn't have done that. Okay, so question 2.
See, voltage activated potassium channels are responsible for the falling phase of the action potential.
Why?
So it's just like the potassium, like they are like inactive, like they leak for a while, and so it allows like ions to seep out. And so I guess like while that happens, yeah, that's what just like brings action potential back to like negative.
Right, but really the answer here is they're asking about those voltage-gated, slow-acting potassium channels. Those are the ones that mediate that recovery phase, the repolarization. And when the depolarization gets to a certain point, those open up and then start to depolarize the cell again.
And then yeah, those like just remain like open longer too, right?
They do,
Okay.
yes. And actually, the answer is on CB 2021. So question 3. Now, first of all, we haven't gone through this yet, but did they actually go through like absolute refractory period, relative refractory period?
He did make a good point to point that out, the
Okay.
inactive.
Okay, then this is fair. So go for it.
He made a good effort.
So who's gonna do question 3?
I will. A second action potential cannot be evoked for a brief period of time following a first action potential. This absolute refractory period occurs because C, sodium channels inactivate.
Yes, but why is that the key factor here?
Because when it's inactive, there's a plug in the channel, so no more sodium can get into the cell to depolarize it.
Exactly, that's exactly the right answer. Okay, so that one was pretty straightforward. Who wants to take question 4? All right, and I will tell you right now, this is a tricky question. It's assuming that you're only working with the information you have in front of you and you have actual no working knowledge of physiology. So just approach this as a Nernst equation question. So answer number 4, somebody. Read it out and answer it.
Alright, I got this.
Okay.
A patient presents with acute kidney failure that results in an abnormally high level of potassium in the extracellular fluid, i.e. hyperkalemia. One consequence of hyperkalemia is an irregular heartbeat and reduced cardiac function. Although the change in cardiac function probably reflects effects on several tissues, you suspect that some of the problems result from a change in the resting membrane potential of cardiac ventricular muscle. Under normal conditions, ventricular muscle has a membrane potential equal to the nerve's potential for potassium ion, equal to 94 millivolts. Hyperkalemia would. I haven't seen these before, so.
So the Nernst equation with relationship to really any ion, but particularly potassium when it comes to excitation, is totally dependent on the relative values intra and extracellularly. And we know that potassium is really high intracellularly.
Right.
It's very low extracellular concentration. So if you raise the extracellular concentration, you're going to alter that relationship by factors of natural log. So if you have a really big difference, then the resting potential is about minus 94. If you narrow that, what would it do?
So by increasing the value
Outside the cell.
outside the cell,
Which means that the ratio is going to be smaller.
you would depolarize the membrane.
That's absolutely right. That's what they're asking here, and they're only asking it relative to the Nernst equation. In reality, what happens is when you get hyperkalemia, initially you depolarize the ventricular muscle, which makes it hyperexcitable. But then you get plugged channels and everything becomes almost unexcitable. So initially, the initial phase is depolarization and hyperexcitation in ventricular muscle, but I think a lot of you are going to medical school, and one of the characteristics of hyperkalemia is an incredibly slow heart rate, like beat, beat, beat. And it's because in response to this hyperexcitation and depolarization, a whole bunch of sodium channels close and stay closed until the hyperkalemia is resolved. So basically the body chose between like tachycardia and bradycardia, and it goes with bradycardia. So anyway, that's the tricky bit of this question. If they are talking about Nernst equation, just think of it in terms of relative ratios and how those would affect excitation. So, all right, so who wants to take 5?
I didn't like this one. I'll take 5.
Okay,
So you have expressed a mutant EGF receptor, EGFR, in a cell which the endogenous EGFR has been knocked out. Mutant EGFR has lost its kinase activity. Treatment of the cell with EGF most likely results in loss of trans-tyrosine phosphorylation of the receptor.
so at its heart, what this question is asking is what kind of a receptor is the EGF receptor?
Tyrosine kinase.
Right. And so if it's lost its kinase ability, what are we know that they're one transmembrane unit, they dimerize, and that once they dimerize, they cross-phosphorylate. Well, it's not going to affect the fact that it's got one transmembrane unit. It's not going to affect their ability to dimerize. So then what's left? Those are the 3 characteristics, the prime features of tyrosine kinase receptors.
So loss of the ability to phosphorylate.
Yeah, to transphosphorylate, yeah. And this is a little tricky because I don't think they ever really discussed with you that the cis phosphorylation or self-phosphorylation is actually mediated at a different site of action than the transphosphorylation. You have to know that to really know that this would be obvious. At least, I didn't listen to your lectures, but at least in what they gave you to read, they didn't really emphasize that. So, all right, so 6. Who wants to take 6?
I can.
Okay.
Platelets at the site of wound release platelet-derived growth factor, which can
bind to and activate fibroblast cells at the site of the wound. When a cell releases a signal used by a nearby cell, the signal is paracrine.
Right, because autocrine is self, paracrine is neighbor, endocrine is you way over there, I might have to give you a phone call, right? Okay. So that one was a give, yeah. That's just like, did you pay attention to this at all? All right, so question 7. Is this the one you had a question about, GQ? All right, this one actually is, I'll go through this one then. So you find that the cells you've been working with express the thromboxane receptor. If you stimulate these cells with thromboxane, it activates the GQ, which tells you if it activates the GQ, this has to be a G protein coupled receptor. Otherwise, it wouldn't. So that one is a given. So if it's GQ, what is that cascade? Phospholipase C, so you have 2 products. You have IP3 and DAG. IP3 activates calcium release at the SER, and then DAG does. So if you look, A is wrong because if cyclic AMP is not generated, that means it's not a GPR, which they just told you that it is, or that it's GI. That doesn't make any sense.
Wait, what? What do you mean GPU?
It's if it activates a GQ, it has to be a G protein coupled receptor.
But it doesn't have to make or activate adenylate cyclase, does it?
No, okay.
As GQ?
Because it's GQ, so A has to be wrong. A would indicate if it's cyclic AMP is not generated, that means A, it's not a GPR at all, or it's a GI or GQ. Well, I guess, but GQs don't have anything to do with cyclic AMP.
So I think I interpreted that as the correct answer.
Me too.
No, I think what they're trying to get you to say is, okay, it's a GQ, it must be a GPR.
I think we're saying
Since it has nothing to do with,
Yeah.
yeah.
You know what? You have an argument there too.
Yeah.
So I think what they wanted you to do here is go through the thought process that A means they wanted you to recognize that it is a GPR. You know that IP3 is formed, so that's not right. And then the level of the receptor on the plasma membrane decreases. What does that have to do with anything? That's not part of what we learned. Cells do not activate G proteins. Well, they just told you that it's a G protein coupled receptor. So, what the heck?
We're getting there. See my anxiety level keeps
Yeah.
getting
And so GQ activation is long-lived. I mean, I think you could actually, on this one, if you wanted to, you could argue A or E.
I picked A because.
Yeah.
Yeah, and there's an argument for that as well. So, who wants to take 9?
But I still have a question. I'm still there. So, can you explain E for me? Like what?
Yes.
Yeah.
Okay, so because the G protein coupled receptors that are activated through GQ actually activate multiple pathways, they're actually slightly longer lived than the others, but they may not have told you that.
Yeah, I don't know that it was explicitly said. And I only kind of interpreted it from like smooth muscle having slow contractions. But I also, what was going through my head was that the receptors in your eyes are G proteins, and those are knocking off like hundreds of signals a second.
Yeah.
So why? I don't know. I was frustrated.
No, you could make an argument. In fact, if you get questions like this on your exam, argue them.
So like,
if you bring them an intelligent argument, they will probably give you the point.
so do they like across the years, like let's say like they had to enter in like maybe they did a bank of like
Possible.
questions.
And this is
And
where
then
things
they
get
enter
awkward
them.
because I have
But, like-
no idea. Because
Well, no,
I
I
don't know
know.
the old tests.
But like, so there's a chance. So for example, what our professor just said was like, well, anything I talked about in class, but like you don't necessarily need to worry about the syllabus stuff that I didn't talk about in class. But then he could ask us questions on the syllabus without remembering that he said that. Like Like, what do you trust? Nothing.
Yeah, it's
No
hard.
one. Trust no one.
Like, really? That's what it feels like. It's a Hunger Games out here.
Okay, I have one more thing on that one.
Thank you. Like,
So the way I kind of justified it after, I found out the right answer, was that, so if you look, it says there's like a bunch of different G alpha proteins expressed in each cell. So would you kind of assume that there might be, G alpha stimulatory and G stimulatory and GQ proteins on the same cell, so you couldn't really say cyclic AMP wasn't being generated because some other process unrelated to contraction could be activating it? Or would you assume this GQ protein on the cell is like the predominant type of, so you've like pretty much just got GQ or GS?
No, you've got them all.
You
You've got them
do
all
have
in
them all,
every
so you
cell.
couldn't really say cyclic AMP
No,
isn't
they're
being
just saying-
generated because it might be something else.
Right, but what they want you to focus on is if you stimulate these cells with thromboxane, which activates GQ,
Yeah.
just narrow your focus. So, A, basically they're sort of implying that either it's a GI, which they just told us is a GQ,
Mm-hmm.
or that it's not a GPR, they just told us it's a GPR.
Mm-hmm.
So, and IP3 is not formed, that's not correct, because we know if you activate GQ, you do get IP3. So that would imply the correct answer would be a GS or a GIO, which it isn't. They just told us it's GQ. And then on C, the levels of the receptor in the plasma membrane decrease. Well, they may or may not, but that has nothing to do with activation of GQ from what they've told you. And so the cells do not activate G proteins, they just told you that it does, so that's just dumb. And then GQ activation is long-lived. So pretty much, you could fight for A, because that's rational, but if you eliminate A through D, then you're left with B, which is true, but apparently they didn't tell you that, which is rude.
Agreed.
Okay. Whew, okay, so you guys have survived the trauma. Let's move on to 8. Actually, we just did this one.
Yeah, this is pretty straightforward.
Yeah. So if it's phospholipase C, then you're gonna get IP3, you're gonna get DAG, so that goes directly to protein kinase C. So basically, by going through the whole process of 7, you've already answered 8. Okay, so somebody jump in and do 9. All right, somebody has to, jump in.
I'll do this one again.
Okay.
Glucose is the primary source of energy for neuronal metabolism. Which of the following is a mechanism that can be used to transport glucose into neurons? And that would be facilitated diffusion.
Right. First of all, passive diffusion doesn't make sense because it's too big, right? And I didn't even look at anything else because facilitated diffusion, there's like 3 or 4 different kinds of facilitated diffusion for glucose, and they specifically went through at least 2 of them for you. So it's facilitated. Let's see. Okay, this one's a give me, so somebody can jump in and take 10 because it's kind of a duh. The only thing they did there is they used a slightly different terminology to describe this. So.
Is it ever called that?
All right, so biotransformation, that makes no sense. Enzymatic inactivation, that doesn't make any sense. So most of these don't make sense because they don't happen in this in the synaptic junction. And we know that norepinephrine is essentially, it's recycled, right? When the cell grows all green. So neuronal transport into the presynaptic terminal means if it doesn't interact with the receptor on the postsynaptic membrane, then it's resorbed by the presynaptic membrane. So that's reuptake. They didn't use a very elegant way to describe it, but that is another way of saying reuptake.
Oh, sorry, I have a question. I was talking about sympathetic adrenergic junction. I've never heard it called that. But my question is enzymatic inactivation by monoamine oxidase.
Mhm.
That is an answer, right?
No, because it's not the major way.
Oh, okay.
It's not the major way that
I was
is
like,
the.
it's totally an answer.
The sympathetic adrenergic junction is your neuronal cleft. And monoamine oxidase always happens within the cell, not at the cell surface.
Okay.
See, so all these other pathways do happen in different ways, but they're not happening right at that neuromuscular junction or the synaptic cleft or whatever, right? That's why. It's sort of like, do you know, first of all, do you know how norepinephrine is inactivated at the junction? And do you know where these things are happening within the cell?
Okay.
That's on page 21. 5.
May I ask a clinical correlation question?
Uh-huh.
MAO inhibitors, the antidepressants, that's exactly how that works. It functions to block that enzyme, so whatever serotonin can't be degraded, or whichever.
Yes, that's right. But again, that's not really it's weird, but that's not actually occurring at the synaptic cleft. It's actually it's altering some of the autocrine feedback mechanisms within the nerve cell itself. So, but that's way more in depth than you're expected to know at this point.
Okay.
So they're asking really tricky questions. I think they're trying to get you to stumble, but just
They did.
yeah.
Success.
Yeah, it's really good.
Okay.
Fell in the mud and everything.
Yeah. All right, so this is a basic, number 11 is a basic question that addresses, do we know parasympathetic versus sympathetic and how they arise at slightly different levels? So we know that all the preganglionics are spinal, and the parasympathetic are, it's called craniosacral. They arise either at the brainstem or cervical area or in the lumbar area of the spine. And then the sympathetic is basically thoracolumbar. That's all they're asking in this question. And so the ones that would be innervating the heart would come from the upper area, and the ones that are innervating like the rectum, the bladder, those sorts of things would be from the pelvic area. And I don't know if they emphasize that or not, but one is lumbosacral, the other is considered mostly
They went over that, right?
We've outlined the specific cranial nerves.
Okay, and then the next few are basically, they're kind of ones that Gina probably handed you, because Gina's awesome like that. So basically, they're just asking what receptors bind to collagen, what receptors bind to von Willebrand. So collagen is glycoprotein 6, and von Willebrand is glycoprotein 1b. So the answer there is B. And then the lab test, this is one that we sort of asked about, but I didn't break them down into extrinsic and intrinsic, and I don't know if she did either. She did, okay. So the answer there is D. So one is intrinsic, one is extrinsic, the other is intrinsic. And then 14 is kind of interesting. So does somebody want to go through the rationale behind question 14?
What did you say 13 was? Sorry, it's like, I didn't.
Yeah, 13 is D. D is Central.
So a good way to remember that one is, so she said PT, PT is quicker, and the extrinsic pathway is much quicker than the intrinsic. So you can just know from there that PT is extrinsic and PTT is intrinsic because it's a longer time for the test.
I'll take 14.
Okay.
So, what is a caveat to using cyclooxygenase or COX inhibitors, such as low-dose aspirin, to prevent the aberrant thrombus formation? I chose C. So we were talking that inhibits thromboxane production, which actually
which is good,
right,
but
but also is like negatives to negative in a sense. So
Right.
the body's inherent way to anticoagulate, it also inhibits that as well.
exactly. And that you just look at that little pathway, COX inhibitors stop here and prevent the formation of arachidonic acid that leads to two different pathways. So when it's one of the things it produces is thromboxanes, one of the things it produces is the prostacyclins. So, and here she's emphasizing again her 2B, 3A, and that's the one that builds bridges. So the answer there is going to be E. Okay, so then I thought things got a little messed up because I think the answers to 16 and 17 are wrong.
I got both of them wrong. I will say that.
And what's the answer? 17. I would think.
So-
I would think C.
Why don't you guys go through 16
that
and
one's
tell me
17.
why
Right.
you chose the answer that
That's
you
not
chose.
right.
Well,
It said that smooth muscle doesn't have troponin. It has calmodulin.
I put calcium because,
Yeah.
Calcium makes sense. ATP does not make
On
sense.
Tuesday, one of the professors was saying, like, if you have a bunch of calcium and it doesn't go away fast enough, then you just stay.
That's what he said
stimulated.
today, too,
So-
with calcium. Like a lot of free calcium or stuff like
Is it
that,
A?
it results in
So-
lower contraction.
Without having with me. so-
Repeated stimulation.
Okay, so number 16 is repeated stimulation of a skeletal muscle fiber causes excessive titanic contraction. I'm pretty sure they meant tetanic.
Yeah.
Okay.
I don't know how many letters are even right.
Titanic's a boat.
I was going with it.
I was like, really big.
I was going with it. All of a sudden I'm hearing songs and, whatever. Okay, so repeated stimulation of a skeletal muscle fiber causes tetanic contraction because the intracellular concentration of what's solute increases and remains at high levels.
Yeah, ATP makes no sense.
ATP does not make any sense. It would be
No
the
sense.
opposite. You would use up all your
Yeah.
ATP. It would drop.
Yeah.
And then you couldn't dissociate.
Yeah.
Am I wrong?
No, yeah, that one makes no sense.
Right.
It has
So
to be calcium.
I think the answer is wrong. And if you get a question like this, if you go and present
That was why I was asking about the tetanus.
Yeah, exactly.
Yeah.
Yeah.
Oh, because of this?
Yeah, because I was like, can we go over what is going on there? Because apparently I didn't get it at all.
Because if you get repeated stimulation, you're going to be opening up the sarcolemma in the muscle cell fiber. You're going to get tons of calcium, tons of calcium, tons of calcium.
Yeah.
Right.
Yeah.
That's what they said.
Yeah. This
Okay.
question is bogus.
This question is bogus, and if you get a question like this, form an intelligent discussion.
Maybe there's a
Said mom.
typo, just like Titanic was.
Yeah.
Yeah.
Yeah, right.
Yeah.
Yeah, if you can't get the letters in tetanic right.
And so I think that 17 is wrong.
It is.
Okay.
Well, oh no, yeah, never
Yeah,
mind.
you went over today that there is no
Yeah.
Yeah, I was
troponin.
confused.
All right, so the question
What was the
is,
correct answer? Or
number
allegedly
17,
correct?
which characteristics
Oh,
or
allegedly
component
B.
is shared by skeletal and smooth muscle? So the first answer is thick and thin filaments arranged in a well-defined sarcomere. Smooth muscles don't have sarcomeres unless they're in the heart, so that doesn't make sense. Troponin, the thing is, I think that's the answer they said was right.
Yeah. And smooth muscle doesn't have it.
Yeah, that's the
But
one.
that doesn't make sense, because yes, they both have troponin, but the I mean, the defining characteristic of smooth muscle is it doesn't have troponin C. It
Yeah.
has
Yeah.
Oh, that's what it is.
Right.
Yeah.
Yeah.
That is one
I
of
would
the
just learn
defining
today. Don't need to
characteristics.
know these.
Maybe it was-
numbers.
Maybe it was supposed to be is not shared.
Then-
Oh no,
but
no,
what,
yeah,
yeah.
no, E wouldn't. I don't know. There.
Yeah.
See how they exist.
Right.
Yeah, and so skeletal muscle and smooth muscles do have pacemakers, so I think we'll be learning about that in the next section. So spontaneous depolarization of muscle membranes, well, I'm certainly hoping that my skeletal muscles are not spontaneously depolarized. I think that would look like a seizure. And a high degree of electrical coupling between cells, well, we know that's not true, right?
Right.
So to me, the answer here is that both are reliant on intracellular calcium to basically undergo their action. So I think that C is correct, and I think that the troponin thing makes absolutely no bloody sense at all. So
Agreed.
again, I would present an intelligent argument and fight.
Should we email Dr. Voigt with these couple of discrepancies?
Yeah.
I tried because on the examination I was given, I found answers that I thought were just blatantly wrong. And I could even cite, like, in the text where it says,
Something
this
different.
answer is wrong.
Yeah.
And he was just like, yeah, so.
No big deal.
So if you get a test answer, if you really feel like you can present an intelligent argument, you know, based on this in our textbook, this answer is not correct.
I'm assuming we can bring all our textbooks when we go, because like we can only look at it like in, like we can go to look at our tests, so can we bring like all of our books, that way like we can argue?
Oh, I'm sure. Yeah, but after you've taken the test, yeah,
Yeah.
You should be able to take your book in there too.
Yeah,
Yeah.
I agree. And then I thought 18 was easy. So skeletal muscle contraction is triggered by A, activation of calcium channels on the muscle membrane. Right, so we're talking about, you have to focus on the word triggered. Triggered, what initiates it.
Mhm.
So B is generation of IP3. That would indicate that skeletal muscle contraction is related to a GPR. We know it's not. C is activation of postsynaptic nicotinic receptors, which we know it is because it's mediated through acetylcholine through nicotinic receptors on skeletal muscles. And it's not part of the sympathetic or parasympathetic nervous system, but Mother Nature, when it finds something it likes, it reuses it. And so nicotinic receptors are still employed, but it's not through a two-synapse method. It's just a direct one axon to the skeletal muscle to a nicotinic receptor. And then D is a conformational change in the troponin tropomyosin complex. Yeah, that does happen, but that's not the triggering mechanism. And then release of calcium from the sarcoplasmic reticulum. Yes, that happens, but that's not the initiating mechanism. The initiating mechanism is the release of acetylcholine from the axon terminal and onto the postsynaptic membrane and its interaction with a nicotinic receptor on the postsynaptic membrane that is actually lying on the skeletal muscle. So that's that. So let's do a quick review. We haven't gone over the last two sections. So ion channels and muscles. So which did you do muscles and ion channels or ion channels and muscles? Doesn't matter.
Channels then muscles.
Okay, let's do channels. So this is CD 2021. Why don't they just have stupid page numbers that are sequential? Really? Come on.
That's so.
You know, I just like things to be organized and logical. It pains me. Okay, so the first thing that occurred to me when I opened up to this particular page was that we have already encountered an ion channel. And what's the ion channel that actually immediately before this chapter, you guys discussed in depth. Right, we talked about nicotinic, which are ion channels, muscarinic that are GPRs, adrenergic, alpha and betas, which are all GPRs. So the channel that you've already discussed that you know and are familiar with are nicotinic. They're ligand-gated ion channels. So, I mean, basically, they're just telling you what an ion channel is. It really is important. Some of these things are not emphasized so much in your upcoming exam, but you're going to have to know the shit out of them if you're going to understand cardiac physiology at all. So the first one is selectivity and the other is gating. So selectivity means that certain channels will only let through not just positive or negative ions, but positive or negative ions of a very selected size. So if they get to be too big, they won't let them through. The ones we already know about, we know about sodium channels, potassium channels, chloride channels. Mostly we've talked about calcium channels in relationship to intracellular events, but there are some actually on the surface of the cell as well. And the second important thing is that they don't just necessarily remain open or closed. There's always a resting on open, closed, and open, and closed. And then under stimulus, they may open and stay open for a while, or they may become closed. And we actually know about this because you guys have just learned about some of the phases of activation, like the slow-gated potassium channels and the sodium channels that facilitate rapid depolarization of the cell, and the potassium channels that facilitate repolarization in a slower manner. And that is all about gating. Under certain conditions, they open and they stay wide open until that congestion changes, and they go back to closed, open, closed, open. But the gating is the activation to close them for a period of time or open them for a sustained period of time. There are lead channels for almost everything. The lead channels for potassium actually are pretty weak. For sodium are actually pretty weak because those two actually form that electrochemical gradient that makes cells excitable and able to respond. What's really interesting is that the lead channels for chloride, which they don't really talk about here, are pretty much always open. They're just kind of open, which is why really alterations in chloride don't really do a whole lot one way or the other to excitability. But alterations in lead channels for potassium and sodium can have huge implications on excitability. So we got lead channels, we got the voltage dependent ion channels. We know about, you guys have just learned about those in relationship to activation, those slow potassium gated channels and the fast sodium. Ligand gated ion channel, that's exactly what you guys looked at in the autonomic nervous system. Those are the nicotinic receptors, whether they be part of the autonomic nervous system or on skeletal muscle. And then epithelial ion channels, these are really fascinating, mostly with relationship to disease, particularly cystic fibrosis. And that is one of Dr. Egan's favorite topics, so I'm pretty sure he talked about that quite a bit. It is kind of fascinating though. So, I think the rest of this is pretty much just straightforward. You need to be pretty familiar with the whole concept of there are chemical driving forces and there are what is it called? Electrical driving forces, and I have it highlighted somewhere. One is based on charge, one is based on concentration. So
Some of this figure 6
Chem-
people
chemical.
around.
Yeah, so I think the
Yeah.
chemical driving force is about
I think it's the next page. of it.
concentration, and the charge one
Right
is,
there.
I think they're called
No, it's
electrochemical
no,
gradient.
It's a page in between that, when your pages are set. There you go. Right there.
Okay, yeah,
Figure six.
yeah, chemical driving force is concentration, the electrical driving force is charge. So, yeah, this little cartoon here at the top of page 4, you won't get a perfect chemical equilibrium because there's gonna be an attractive force for some of those positively charged hydrogens to stay with the negatively charged ions, and that is actually exactly what happens in the cell. There are lots of negatively charged proteins that cannot go in and out of the cell, and that is one of the reasons that the cells actually will tend to have a higher concentration of positive things like, you know, potassium or sodium. So, excitable cells, for example, what's a classic excitable cell? A nerve, perfect. Tons of permeability, potassium. Potassium. That's it. So at rest, if we're talking about leak channels, we know that potassium is generally high within the cell. So a leak channel